Hi, welcome to this video where we're going to be looking through the second paper that you did for your end of year biology exam. So that's the practice paper, Depth in Biology. So once again, really well done. In fact, I think the marks for this second paper were even better than the first paper. So fantastic work. Where people lost marks, I think, was often the essay questions. Now, it's kind of about strategy. So the most important part of these videos, I think, is really to understand the marking of the essay questions and understand the strategy for doing those. Because often you said some really good stuff, but because you didn't talk about all the things the question needed you to do, then you might have had a maximum number of marks that you could get out of 6 of like 2. So you could, even though you said loads of good stuff, you could only get 2 out of 6 because you missed one aspect of the essay. Okay, so we're going to dive straight in. Here we go. So question 1. Evolution is the change of species over time. Here are 2 organisms. Explain how scientists are able to estimate the age of extinct organisms. So the first point is this idea. The idea that you can look at the layers of the rock, so fossils, layers of the rock, and that the oldest layers are deeper down. Now, you could have said radiometric dating, but not carbon dating. Why, you may ask? Well, carbon dating only has a useful range of 100 to 30,000 years. So if we're talking about in the millions of years, it's not going to work. So we have to use other isotopes. So that's just something to remember. Didn't really go into that much detail about that in the syllabus, but that's something else for you to know. Carbon dating only works in the sort of thousands of years range, but not the millions. And then let's do this one too, before we look at the mark scheme. Identify 2 features visible, okay, visible in both species that suggest that species A and B evolved from a relatively close common ancestor. Well, there's loads of things. You could say segments on the thorax, for example, or you could say maybe lateral spine. The anterior spine, no, because that one doesn't have it. So segments on the thorax. I would probably say that as a segmented thorax would be the best way to say that, and lateral spine. Okay, let's check the mark scheme here. Scrolling through. Okay, so actually the marks here, the first one, the 1 mark for using the word fossils, and then the deeper in the ground idea. And now you could have get radiometric dating, but not carbon dating. Similar tail, segments, lateral spines, other different things you could have said there. But you only got 1 mark for saying both things. Okay, on to the next page. A student used figure 1.1 to make a number of observations comparing species A and B. One such observation was that species B had longer lateral spines than species A. Now, you can't say that. And the reason is there is no scale on this picture. So we don't know, in fact, if species A and species B are the same size. We don't know if they're the same scale. We could say that relative to body length, species B has longer lateral spines compared to the length of the head, thorax, and tail, whereas species A has relatively shorter lateral spines relative to body length. But you can't make an absolute judgment about that. So the mark scheme says exactly that. So we're looking right here. So let me zoom in a little bit more. That's better. So first of all, we've got the idea that there's no scale given. That's worth 1 mark here, no scale. And the idea that you can't compare absolute length. However, you could also get a mark for saying that you could compare relative length. Might be a valid comparison. We also don't know, here's some other things you could say, we also don't know that how old the specimens are in terms of their maturity. So one specimen could be a year old, the other specimen could be 5 years old. So it might have time to grow more fully. And also, we've only got 1 of each species, so we don't really know what kind of variability there is in lateral spine length within a population of these extinct organisms. So those are all some things that you could have said. Here we have the diagrams here, and it asks you for an adaptation and an explanation. Some people went a bit off here because they didn't quite spot something key in the question. Well, Often in questions like these, it only wants you to talk about things that you can see in the diagram. This question, it doesn't really highlight that well, but the mark scheme is that. So it says identify and briefly explain an adaptation. So it's really the key is in this word identify, because you can't identify an adaptation from the diagram that you can't see. So if you talked about spiracles and tracheal system, the internal kind of parts of the organism that you can't see, then you can't get marks. Okay, so what can you see? Well, you can clearly see a tail, which is for swimming. So that's one thing you could say. And you could clearly also see segments here and here, which allow a flexible body to kind of swim underwater. So those are the things that you could have said. And here's the mark scheme just to confirm that. We're looking here. Adaptation and explanation must both be present to award the mark. So tail for swimming and movement, or segments for flexibility or swimming. Right, this one. It's a 4-marker, okay? So that's important, 4 marks there. You want to say 4 things. It says, explain how biological molecules can provide evidence that species have evolved. Then it goes on to say, giving you clues, massive clues, you should refer to different types of molecules in your answer. Okay, so what different types of molecules can we use? Well, I think most people are aware that we can use DNA. Is there another type of molecule that we can use? Yep, we can also use proteins. Okay, so here are the marks. So DNA, that's kind of worth a mark. And then how do we use DNA? Well, we have to sequence it. And you really have to say that we compare the sequences, and the degree of difference indicates the degree of time since the organisms have shared a common ancestor. So sort of difference in sequence equals roughly kind of a measure of evolutionary distance, you could say, between the two organisms. We can also look at proteins. And really what we do there is we look at the amino acids sequence in shared proteins. For example, if we're looking at primates, primates have hemoglobin, all primates have hemoglobin. And we can look at the amino acid sequence of human hemoglobin versus, I don't know, let's say ring-tailed lemur hemoglobin. And we can work out the number of amino acids that are different, and that gives us an indication of when humans and ring-tailed lemurs last shared a common ancestor, which is about when Madagascar separated off from Africa. Okay, so let's check the mark scheme there to fully check that. Okay, so the biological molecule, first of all, we could talk about nucleic acid or DNA, or mitochondrial DNA is what that stands for, or RNA. So that's the first mark there. And then this idea about the difference is how different the sequences are. But you really have to say that sequence similarity or the sequence difference. So if you didn't talk about sequencing, then you couldn't get that second mark. And then for the proteins, or you could have given an example of hemoglobin or cytochrome C. Many of you talked about that. Well done. And again, it's the amino acid sequence down here. Amino acid sequence, you had to say that. Or you could have said the primary structure, because primary structure is the sequence of amino acids. And tertiary structure and stuff like that, no, that's not what you want to talk about. It's the amino acid sequence or the primary sequence or primary structure. Okay, here we have some statements that Charles Darwin sort of thought about, his observations that he thought about. I'm zooming in a little bit. Okay, so he used these sort of observations to deduce ideas. So first, his idea is this: characteristics are passed on to the next generation. Which of these back it up? And the answer is E. Okay, just E. Offspring generally appear similar to their parents. Next one, there is a struggle for existence. Not everything can exist. So this is going to be G. Organisms have the ability to produce large numbers of offspring. And it's also going to be H. populations in nature tend to be relatively stable, because if you're producing large number of offspring, but then the population isn't growing, that must mean there's a struggle, because some offspring are surviving and some are dying, so there's a struggle for existence. G and H together struggle for existence. And then the last one, individuals' beneficial characteristics are among the few who survive. This is kind of F, G, and H here, because all these three, we've got the aspect of struggle plus the aspect of individuals not being identical. So that means that the ones that are best adapted survive. Double check the mark scheme. E, G and H, and F and G and H. You need both letters here and all three letters here for the mark. And it's 1 mark for each row, in case you were wondering. All right, we'll skip past this blank page. And on we go to this one. This question 2 is all really about biodiversity. Okay, so we've got an area accessible to walkers, an area fenced off, 2 different mornings. Now this is, I think a lot of people just misinterpret this sentence. It's not a great sentence, to be honest. And I think in a real paper, because this was a practice paper, remember, they do kind of check for understandability, if that's a word, a bit more. So this says, on 2 different mornings in June, the students walked along a transect in each area 4 times at 30-minute intervals and recorded every butterfly sighting. So some of you interpreted this to mean that, like, they walked, you know, I don't know, let's say on a Monday, they went and looked at area 1, and then on Tuesday they looked at area 2. Maybe that's true, but we don't really know. Others of you interpreted it to mean that, like, Monday they walked area 1 and area 2, and then also on Tuesday they walked area 1 and area 2. So kind of difficult to say which they meant from the sentence. So it's not a great question, to be honest. But how could it be improved? I'm just going to go straight to the mark scheme. Things that you could have said. First of all, more transects. Okay, more transects in different parts. We had to say in different parts. I don't think you got a mark for just saying more transects. More transects in different parts of area 1 and 2. So, you know, area 1 and 2 are big areas, but you just walked in one little area, but you have to take your transects in various different parts of area. Maybe you only looked at the bit of area 1 that was like a field, but you didn't look over near the pond, and therefore it's not very valid. Secondly, different times of day. In both places, they looked at a morning, so different times of day might have given you different results. Maybe some butterflies only are visible at night, or in the evening, potentially. Different times of year, because we only looked in June, so these are all kind of ideas that to do with sort of the variability of the day. Then there's this idea of a method to ensure that individuals are not counted again, because you're walking along, you might count the same butterfly 3 times, and that's not fair. So potentially something like sweep netting might have prevented you from counting twice. So this also would have been a method to capture individuals. And use a method to correctly identify the species, like a key. I think even someone said an app off the back of hopefully our biodiversity using the plant app. Yeah, there are probably apps for identifying insects. I'm sure there are. So those are all the things you could have said. Maximum 3 marks. We're going to come back to this table because we're going to use it to calculate Simpsons index biodiversity, but we're going to do that just in a second. So identify the area with the highest species richness. Well, species richness is a measure of the number of different species, and I think everyone got this right. Everyone said area 2, because there are more species. There I've got 1, 2, 3, 4, 5, 6 species present. Because if we look over here, this species is not present. The silver-studded blue is not present in area 1. So area 1 only has 5 species. And that's what you need to say. Identify the area with the higher species evenness and justify your answer. Now this is probably the more difficult thing, and justify your answer, not just say because it's obvious, give a reason. Well, the area that has the better species evenness is, again, area 2. So why? How do we explain that? Well, if we look at the range in number of individuals, we've got a range of from 2, which is the lowest, up to 11. So that difference, that range here, is 9. Okay, we've got a range of 9. And here we've got a range of, a larger range. I guess we've got a range of from 2 to 16, so that's a range of 14. So the range in the numbers of individuals is greater. What you don't get marks for is just repeating the question. So here's what I mean. I'll put it in different blue, so this is not worth of a mark. Some people sort of wrote because the species are spread more evenly, but that is basically just saying because it's got a bigger evenness. So this would not get a mark because the species are spread more evenly, because it's repetition of what is already told to you in the question. Right, now we've got to do the Simpsons diversity index here. Now, I think the people who didn't make any mistakes, often they use the table. The table's above, and it's kind of already laid out to help you. And in fact, it even says, it tells you to do this. Look, you may use the blank spaces in table 2.1 to assist in your calculations. Good idea. Let's do what they've suggested. Okay, so I'm going to do this calculation up here. Now, the first thing we need to do is we need to calculate big N. So let's work out big N, which is the total of all these things here. So let me just erase this. So if we total up all these here, we're going to get big N. I'm just going to type away here, and I'll edit this video to speed through the calculation so you can check what I'm doing. Okay, so I total that up. Big N is equal to 40. All right, so the next thing we need to do is we need to do little n over big N. And then in this final column, we're going to do little n over big N squared. Now, I would keep, lots of numbers of decimal places. The reason you've got to keep lots of numbers of decimal places is look over here. This is to 4 decimal places. So they're going to want you to calculate it, your version, to 4 decimal places, which seems a bit much to me, like a lot much. But we've got to keep consistency with the numbers that we're being given. So that's a key message. Right, I'm going to be quiet. I'm going to go through the calculation and you can see how I do. Okay, so there's the first column now. I've got to square each of those values. Okay, full disclosure, this is the second time I've done this question because I made a stupid calculator error somewhere in there first time round. So it's so easy to do. So really feel for you if you did the same thing as me. I put that in blue, the error that I made first time around. I did this bit wrong here. So that is the right number. Those are the squared values. This is my error again. And then you should have got this D equals 0.7975, which was equal to 4 marks. The mark scheme is you get 3 marks for the number, but you have to get the 4 marks, you have to have it to the right number of significant figures. So answer given to 4 significant figures, and I did accept 7975 instead of 7976 because that comes from including the decimal, including sort of up to 5-6 decimal places all the way through your working. So either 0.7975 or 0.7976 are acceptable for 4 marks. Okay, let's move on from that horrible question. C. All right, okay. This is a good one. Students concluded that fencing off the area of heathland has increased the biodiversity of butterflies. Worth reading that again. They've concluded that fencing off the area of the heathland has increased the biodiversity of the butterflies. That's the student's conclusion. Now, another key thing to look at is this word, evaluate the validity of the student's conclusion. With evaluate. So what does that mean? So evaluate means give some points for supporting the conclusion, give some points against the conclusion, and then come to your own judgment, okay? Supported by your evidence. Now remember, I've said this before, that most of the time, 90% of the time, a question like this wants you to say there isn't enough evidence to support the conclusion. Normally, someone will make a conclusion and it will be kind of seemingly right, but when you go deeper, there's not enough evidence to support. They're actually training you to try and be good scientists here because if you're a scientist and you're reading a paper and you just like glance at it and the scientist says, clearly fencing off increases biodiversity. Maybe it sounds good when you just hear it, but then if you go into the data, you might see that actually their methods are flawed and it's not correct. So that's what they're training you to do. So remember that, bear that in mind. Pretty much they want you to take down this person's conclusion, but give a bit of positives as well. Right, let's talk about it now. Okay, so right, what can we say? Positives. I'm going to kind of mind map this. Let's do the positives first. Okay, so first of all, there is a higher Simpsons index. Is higher. Okay, so that's clear. And also, there is a higher richness of species, one more species, and a higher evenness. So those are all good things. But then in terms of the negatives, I'll do the negatives in blue here. There's loads. First of all, we only looked at it one day. But all the method things that we talked about before, so different seasons weren't considered. Another thing was looking at the data. So this is the kind of method stuff. Only look one day, different seasons, not that many transects, all that stuff. So the method was a bit rubbish. What about the data? Negatives on the data. Well, the two different values of Simpsons index were quite close together. Are they really that different? Not really. So I'll just SI for Simpsons index. No, actually, let's use D. D, Simpsons index of biodiversity, quite similar. So it's difficult to really say if they were significantly different. And another thing is I would call causality and time. We didn't have any measure of the biodiversity of the heathland before it was fenced off. Maybe that part of heathland, due to other factors that we haven't considered, has always had a high biodiversity and always will. And it's nothing to do with the fencing. So causality, no previous record of D for Simpsons index of biodiversity. Those are all some things you can say. Now, let's pay close attention to the mark scheme here. So the mark scheme is this kind of tiered mark scheme. Right, now in a minute you can pause this and you can like really look at all the different points over here. Right, I've talked about some of them, but I probably forgot a few. So here are all the points that you could have said. But you don't just add up the points and that's your mark, okay? You have to look at the criteria of the levels. So let me take you through them very quickly. First of all, 1 or 2 marks. An evaluation of the claim. You have some discussion. You can either talk about the method or you can talk about the implications of the data, and you include evidence for or against the claim, but there may not be a judgment. So evidence for or against the claim. So some people just talked about, yeah, the claim's great, here's some evidence, done. Maximum 2 marks. Right, moving up a level. Level 3 or 4 marks for level 2 answer. So you have to talk about the method and the data, and all the implications of the data. You have to give a holistic judgment, and you have to have evidence for and against the claim. So you basically need pluses and minuses. So you need pluses and minuses, and you make a judgment. So you also have to have a judgment there, and it could be your judgment for or against. But look, to get the full 5 or 6 marks, the judgment has to be that there is not enough evidence. Okay, that's the correct answer. There isn't enough evidence. So you have to make that judgment, and you have to talk about loads of things to get to that point. You have to talk about the method, you have to talk about the data, and you basically make a holistic judgment. And you've got to use lots of detail, full and detailed evaluation. Okay, so if you want to look at that mark scheme some more, you can pause this video here and see where you think you could have improved your answer. And I've also got for you two of your examples to see some people who really nailed this question. All right, this one. Here's a 6 marker. 6 marks, okay? So let's see why. Well, first of all, you can see in my marking here, I picked out 1, 2, 3 positive things that they've said. So they've talked about Simpsons index diversity, so they talked that's higher. So they talked about the comparison. And they also talked about species evenness and richness in area 2 being higher. However, there is not a massive difference, so then this is now a negative. There is not a massive difference between the calculated values, so that's that first negative there. Diversity in area 2 is only that much above. Furthermore, there are a greater number of both, so they've talked about the actual the species of butterflies that are more abundant in the other area, so that's looking at data again. So, and then they also, now they start to talk about the method as well, which they need to talk about the method. So the conclusion may also be invalid due to their collections. So they talk about the flaws in the method. They've only collected data at one time of year, and so on and so on and so on. So they've talked about the data, they talked about the method, and here, right at the bottom here, let's highlight this. Because of this, I do not think there is enough data to support the student's claim. Okay, so they've picked it apart, and they've made a judgment. That's why this gets 6. It's a level 3 answer. This one is great as well. Didn't quite get 6. This was a 5 marker. If you want to have a look at this, pause the video and off you go. You can see the 2 points for the claim, 2 points against the claim, but I didn't quite feel it had as much detail as that 6 marker. Okay. Pause the video now if you want to have a look, and now we're going to move on. Okay, so on to question 3. And I've added a little diagram here of a heart, sort of more of a colourful, sort of more realistic diagram, so we can compare. So the structures labelled A here are the coronary arteries. In actual fact, you don't need to know this, but there's actually coronary arteries, these are the red ones, and coronary veins, these are the blue ones, and they supply the actual heart muscle with blood, with oxygenated blood. So this B here is the cardiac muscle. Now I think on the mark scheme you may have been allowed to have just muscle. Let's just double check. So yeah, so coronary artery and then muscle, or cardiac or heart, either one, muscle. Now I know that at least one person wrote pericardium, which I think is a medical term. I think that is an accurate term, but it's not on the A-level syllabus, so I don't think you would get marks for that, pericardium. So cardiac muscle is what it is. And we're going to look specifically about cardiac muscle a bit more in year 13 when we look at different types of muscle tissue. Okay, scrolling down. Right, here I've dropped in a photo of a heart dissection. Apologies for the slightly graphic picture. But we need to sort of recognize what they're asking us here. So this question says, we've got a mammalian heart, the internal structure of the heart was examined, and then basically it says the atria were removed. So what would that sort of look like? So if we were to remove the atria, we would sort of be cutting sort of through this kind of region up there. Now, I've looked at the mark scheme here. It's not really. This question is pretty tough, to be honest. And now they say that you would still be able to see the atrioventricular valve. I guess you'd still be able to see just the bottom part of the atrioventricular valve. What else would you be able to see? Bundle of His? No, you would not be able to see that because those are internal nerve fibers down the septum, so you can't see that. Left ventricular wall? Yes, you can see that. It's here. That's the left ventricular wall. So yeah, that's a good one you can see. Pulmonary vein. Pulmonary vein supplies the right atrium, so you wouldn't be able to see that. So the pulmonary vein would sort of bring in, I think it could be that one or it could be that one. I'm not sure which one it is, but it would be one of those two. Purkinje fibers, again, no, because those are sort of internal nerve fibers that are in the heart, sort of throughout the heart muscle tissue, so you can't see that. Sinoatrial node, again, there's not really any visible sign of that. It's a region of the heart that sets up the electrical activity of the heart, but you can't really see it. Now, semilunar valve, it might be pretty difficult, but the exam board certainly thinks that you can see it. Now, whether you can see it right here, not really, but remember the semilunar valve is the valve that separates the ventricles from the aorta or from the pulmonary artery. So it's kind of connected to the ventricles. It's associated with the ventricles, so therefore you can see it. And the septum is the thing down the middle, so yes, you can see that as kind of, I don't know where that is here. It's difficult to tell, but it's down the middle. So yes, you can see the septum. Okay, so those are the answers. Now, if you got it all right, you got 3 marks. I think if you got sort of 1 wrong, you got 2 marks. If you got 2 wrong, you got 1 mark. So that's basically how it's scored. Tough question, that. Yeah. Okay, now let's move on. Now, this diagram shows pressure changes inside the heart during one cardiac cycle. Now, some of you actually missed a question. You just completely forgot this question here. Here it is. Draw a line on figure 3.2 that shows the pressure changes in the right ventricle. Right, so the right ventricle, remember, has lower pressure than the left ventricle. But it contracts, the heart contracts both sides at the same time. So the left and the right ventricle are doing the same thing, just the right ventricle slightly less forcefully. So therefore, let's draw on this line. It's going to change with a similar sort of shape to the left ventricle, just at a lower pressure. So here, the left ventricle is this dark line, so it's going to be pretty much the same for there. But then here, it's going to go up, and it's not going to reach as high a height. So it's going to be a bit like that. Okay. So this is a 2 marker. First mark for the same shape, and second mark for having a peak of pressure that is lower than this one. I think it had to be below 15, so below about here, but somewhere in this range. Let's double check the mark scheme there for those last few ones. Okay, so there's, first of all, there's a mark scheme here for that table that we filled out, and here's actually the kind of breakdown of how you get your, out of the 3 marks, how many ticks do you need. And this is the grading criteria for that, the line which you drew on the graph. So first of all, the line clearly rises and falls at the same time as the left ventricle, so basically the same shape, and then the peak between 2 and 15 kilopascals. Yeah. Okay. Now, the next question. 6 marker. Tactics. Okay. Remember I said it's all about the tactics of the 6 marker. So the first thing that we're going to do is make sure we've read the question thoroughly. So, describe the events taking place at the points marked W, X, Y, and Z. Those are all in bold, okay? So you have to talk about each point, W, X, Y, and Z. And if you don't, you're not doing what the question asks you. And explain how these events are related to the changes in pressure. Okay, so clearly we need to talk about W, X, Y, Z, and what's happening in terms of the pressure at all of those points, and specifically not just the pressure, but the changes in pressure. Okay, so let's go through, first of all, the mark scheme, okay, and then we're going to take you through what's going on. So the mark scheme is really specific. And actually, you can pause this if you'd like and have a full look at it in a second. But I'm actually going to take you through the level descriptors, because really this is how you need to start thinking. All right, so for the first one, level 1 or 2 marks, not many marks. So you describe the behaviors of the chambers or the valves. So basically, when you're talking about the valves, you're either saying, are they opening or are they closing? And the chambers, that's the ventricles and the atria, are they contracting? I.e., are they in diastole? Sorry, diastole is the relaxing, or systole, which is the contracting. So are they doing atrial systole or ventricular systole, or the diastole? And you describe the pressure changes, like saying, is it increasing or decreasing at some of the points? Okay, so you haven't talked about all of them. Now, to get to 3 or 4 marks, you have to talk about at all of the points W to Z. So many of you forgot one of the letters. You missed out W. And if you did, you could only get a maximum of 2 marks. That's the first major thing that messed some people up. Then again, what else do you have to do for 3 or 4 marks? You have to talk about the chambers or the valves, and this is an extra bit now, and relates some of these behaviors to changes in pressure. So you have to say how the pressure causes the valves to open or close. Whereas before, you're just sort of saying the pressure's going up, and it doesn't really relate it. For 5 to 6 marks up here, you've got to talk about the chambers, so the ventricles and the atria, are they contracting? You've got to talk about the valves, and you've got to talk about W, X, Y, and Z, all of them, and you've got to talk about the pressure changes at all of the points W to Z. Okay, so you've really got to go for it, full detail. Now, the stuff that you need to talk about in terms of chambers, valves, and pressure is over here, but I'm now going to take you through a sort of model answer for that. All right, so let's start with, I'm going to do it in bullet point form. So let's start with Y. Remember when you talk about each one. So at Y, what we can see here is the pressure, and I'm going to do a little quick color coding. Okay, so now I've done my little quick color coding there. So I've got the aorta in green, the left ventricle in red, and the left atrium in blue. Okay, so what's going on at Y? So we have the pressure in the left ventricle goes above the left atrium. So P for pressure in left ventricle rises above that in left atrium. So we have an increase in pressure. This means that the AV valve closes, and some of you said it closes to prevent backflow of blood. And all this is because we have the start of ventricular systole. Okay, so you can see how I've done the three things. I've talked about the pressure changes, I've talked about the valve opening or closing, and I've talked about what action is happening. So that's Y. Okay, Z. Still in ventricular systole, still contracting, but now the pressure in the left ventricle has risen above that in aorta, which means that the semilunar valve opens, allowing blood to flow into the aorta. At W, now we're in ventricular or just general diastole. So let's just call it diastole because the ventricle systole has finished. If you're wondering where does that finish, by the way, pretty much at the peak here. Okay, so this is V systole, and over here this is diastole, the whole heart resting, so ventricular and atrial diastole. Okay, so W, we've got the diastole, we've got the pressure in the aorta falls below, sorry, no, I made a mistake there. Pressure in aorta, the other way around. Pressure in left ventricle falls below aorta. This means, therefore, the semilunar valves close. And let's not forget X. In X, we're still in diastole, but now the pressure in the left ventricle has fallen below the left atrium, so therefore the AV valve opens, allowing blood to flow from the atrium back into the ventricle. Okay, so I've talked about everything there. That's what you need to do. So hopefully that's kind of pinpointed where you've gone wrong or where you didn't get the maximum number of marks. And now I've also got 2 exemplars, so you can see what some students who really nailed this question, what they did. Okay, so let's have a look at those 2 exemplars if you want to. Okay, so first of all, here's an exemplar, which you can pause and have a look at if you'd like. So this person got 4, but they forgot to talk about W, I think. So they talked about, this is C for chambers, they've talked about a chamber, they've talked about V for ventricles and P for pressure. V for valves. So C for chambers, V for valves, P for pressure. But then here they talked about valves and pressure, but they didn't talk about what was happening at W. Maybe I shouldn't even given this 4. Maybe I should have given this less because they missed one. So this should have been maybe given 2. Sometimes I'm a bit generous. So pause that, have a look at it if you'd like to, and I'm going to show you another one in just a sec. Okay, here's another one. So this one I gave 2. They were really nice. They talked about Y, Z, and X, but they didn't talk about W. And also in this one, they talked about pressures going up and going down, but they didn't compare pressures. They didn't say the pressure in the ventricle is higher than the aorta and therefore that sort of thing. You need to talk about relative pressures. So again, have a look at that if you'd like. You can pause it, have a look through, see how I've marked it. Finally, one more example. This is a 6 marker. This is bang on. Lovely detail here. Just zoom in a teeny bit more. And this is given 6 marks because they talked about, you can see the C, chambers, valves, pressure, chambers, valves, pressure, chambers, valves, pressure, and hopefully chambers as well. So they talked about all letters, Y, Z, W, X, and they've talked about pressure, valves, and chambers at each point. So this is a perfect answer here. Pause that and have a look if you'd like. Okay, let's get back to the other questions. All right, okay, so here we are on the next part of this question, looking at some ECG traces. We've got the ECG from a normal heart, and then after it's been treated with digoxin. So we are asked to work out the heart rate from the ECG. Now, the first thing to sort of recognize here is that you should pick a point that is very easy to distinguish. So the best point to do that is everything around the QRS complex. So in this one, we can look at this point here and compare it directly to this point here, okay? Because that's the start of the same point. So if we then go down here, that is going to be exactly at 0.15 seconds, and that is bang on 1.1 seconds. So then we can work out how long it takes. So 1.1 seconds minus 0.15 seconds is equal to, calculator, 1.1 minus 0.15 equals 0.95 seconds. So that's so it takes just under a second to do a heart rate. So then when we're calculating the heart rate, it's going to be over 60, isn't it? Because if each beat takes just less than a second. So just bear that in mind when you do your next step, because several people went wrong. So now we do 60 seconds divided by 0.95 to get the heart rate. Okay, 60 seconds in a minute, 60 divided by 0.95 is going to be equal to 63.2 BPM. Okay, so first of all, that's the heart rate. Now I need to work out the percentage change. So I almost need some more space here, don't I? So we're going from 75 to 63.2. So what is the actual change there? The number change, so 75 minus 63.2 equals, it's a change of 11.8, divided by the starting value, which is 75, times 100 to make it a percent, so 11.8 divided by 75 equals times 100, so 15.7%. And it is a negative change, isn't it? So it's minus 15.7%. Now let's check. Let's hope I got the marks for this. 14 plus or minus 1%. So I think I would have. No, I've given it to 16, haven't I? I said I got 16. I think that's bang on. I think it's minus 16%. Maybe, okay, so if you got minus 16%, I'm going to give you the mark here because I think this is wrong. But potentially, maybe I've measured it wrong. I don't think I have. Let's just double check that I've zoomed in a little bit more. So I really want to, I'm going to get rid of the scrolls that I did on the outside. Yeah, okay. So that is my measure. This is so, you've got to be so accurate here. Look, if I look here, actually the start of that peak is right there. So that's 0.16 is what it should be. And that is exactly 1.1. So if I was to do that 0.16, that's going to be it's going to change everything, isn't it? And I think that will probably just about give me the right answer. So that would be 9.4. That would be, I'd have to change this. That would be 9.4. That would change, and so on. Okay, so you really got to be accurate. I don't know if I get the marks. I probably get 2 marks out of 3, but you've got to be accurate with your measuring. I tell you what is not a good idea to do is you don't want to measure sort of with one of these hump bits, because that's, they're a bit vague. You don't really know where to measure from. So I would always pick a sharp point to measure from. Okay, moving on. Explain why the answer calculated in part 1 may not be accurate representation of the patient's heart rate and suggest a more accurate answer could be obtained. Well, the first thing is we're only looking at one heart rate, and there are variations, so it would be much better to look at the number of heartbeats in, let's say, a minute and measure that, okay, to get an average or a mean. So let's look at the mark scheme. Let's go straight into that. So we're only looking at one cycle or heartbeat, only one heartbeat. So it could be anomalous or atypical. So idea that measurement, and also the mean would be better. Also, this is an extra one which we've sort of talking about. Idea that measurement of cycle from different points gives different values depending whether you measure at the beginning of the QRS complex or the T wave, etc. Right, on to the next one. Digoxin causes the heart rate to change. And here we go. We have again this word identify, one other effect of dioxidant evident from figure 13. So that means we have to see something on the figure and talk about it. So let's go up. What can we identify? The clearest thing is that this T wave here is longer than the T wave here. So a longer T wave would be the best answer. Mark scheme. Longer T wave, or you could have also said broader R wave. Okay, longer T wave or broader R wave. Okay, moving on to the next question. All right, question 4. So first of all, it asks us why phenolphthalein indicator was able to detect the breakdown of triglycerides. Remember that a triglyceride is basically kind of this shape. We've got the glycerol and we've got the 3 fatty acids. So when we break down the triglyceride, the fatty acids are released and they lower the pH. So that is the answer here. If we look at the mark scheme, the breakdown product is the fatty acid, and you have to say that the pH falls. As more, and that actually I may have been a bit too generous with this one because it says as more breakdown occurs. So I think you have to get the idea of that as the triglyceride is being digested, it gets more and more acidic because more and more fatty acids are released from the glycerol molecules. Okay, so maybe that's just a little extra bit to include when you're looking at your paper result. Okay, now we've got to do a standard deviation calculation. Okay, so this standard deviation calculation will give in the formula. So it's the sum of x, which is the value measured, minus x bar, which is the mean, all squared, over n minus 1. Now, I'd probably go up here and actually use this space. So I'm actually just going to kind of add a row of my table, probably, down here to use this space. So we're already given x bar, which is the mean. So now we need to do x, which is the value, minus x bar. So I'm going to do that. So I'll just edit this to make it a bit faster. Okay, so there's my x minus x bar. And then the next thing I need to do is I need to square those values. And now I need to sum those. So if I add those up, so the sum of all those is equal to 8,700.49. I divide it by n minus 1, so n is 3, so I divide it by 2. And then I square root the whole thing. Equals 65.5 0.95. Let's make that just to one one decimal place. So that would be, if we round that, that's actually going to round to 66.0 exactly. Three significant figures or one decimal place. So write that down here. Now check the mark scheme, and that is okay. 66.0 for two marks, 1 mark for either 66 or any answer between 65.9 and 66. So the sort of second mark is really to give it to the appropriate number of significant figures. Okay, on we go. Okay, next one down. Standard deviation measure of spread results. All significant variables were controlled in investigation. Identify limitation to the experimental procedure that caused a high standard deviation and suggest an improvement. Well, you could have a few. You can just have the one. Limitation is the idea that the endpoint is a bit difficult to tell. It's subjective or difficult to judge. So we're looking for a color change, which is maybe difficult to see. So as an improvement, we could use a pH meter. It does allow some other things. You could have said fat separates to the top of the solution, or and then to keep mixing throughout the experiment because the fat might separate off from the water. Triglycerides contain 3 elements. M and N are formulae for 2 macromolecules. Which looks like a triglyceride? Well, there is a short way of doing this actually, and it's just to look at this. Oxygen 6, that means it's a triglyceride. And actually the ratio of the oxygen to the carbon and hydrogen is way less. If you look here, actually this ratio is very close to CH2O to the N, which means that it's a carbohydrate, and this one is a triglyceride. Whereas this one is a fatty acid. So the idea is that it's M. There's a lot less oxygen, a lot fewer oxygen atoms. The reason there's actually 6 there is because for each kind of ester bond is like that. So there's 2 oxygens per ester bond, and we've got 3 ester bonds. Okay. Phospholipid molecules are similar to triglycerides, but they also contain the element phosphorus as part of the phosphate group. Explain how the structure of phospholipids allows them to form bilayers of a plasma membrane. Okay, well, you need to get a few things here. So let's just draw a quick bilayer. So first thing you need to talk about is head, polar, hydrophobic. I believe in the mark scheme, we'll check in a second, that you also need to get for this mark, you need to talk about the tails, non-polar, and oops, I got those the wrong way around, sorry. That's hydrophobic, philic, hydrophilic, and this is hydrophobic tails. Okay, I think that's the first mark. Then you need to get the idea of the fact that we've got kind of water or an aqueous environment on the outside of the membrane on either side. We can talk about aqueous environment, and we can say that that's one mark, and we can also say that the heads form hydrogen bonds with water outside. So we could even put in a little H2O and sort of draw some hydrogen bonds there. And then we've got in the middle here, we've got the tails face towards each other. To orientate themselves away from water, and there's hydrophobic interactions there. Let's check the mark scheme. Actually, those things you could say. So first of all, you need to get the hydrophilic head and hydrophilic tails for this mark. The second part, the hydrophilic tails repelled. I didn't say the word repelled, should have done that. Repelled by water. Or facing away from, orientate themselves away from. I think that would probably get the mark. The head part forms hydrogen bonds with water. The idea that the medium outside is aqueous, and the idea that the hydrophobic nature of the tails results in their facing towards each other. So those are the marks that you could have said. Next, the plasma membrane also contains proteins which are made within the cell. Outline the processes and organelles involved in the translation of these proteins from RNA. I think let's just go straight to the mark scheme because it's kind of got it in the right order. Can't really make it any better. So first of all, we have to say that mRNA is transported out of the nucleus. You could have said via a nuclear pore, would have been a good thing. Then it goes to the ribosome. It associates with the ribosome, binds to the ribosome, which is where translation occurs. Translation occurs at the ribosome. And then we can get some marks for describing this process. So the fact that the transfer RNA brings specific amino acids into place, or just sort of describing this process. And then we've got peptide bonds forming between adjacent amino acids. And eventually the protein that is produced, or the polypeptide that is produced, can also be processed through the Golgi apparatus and then arrives at the cell surface membrane. People put extra detail in as well, which was great. You were talking about the fact that the RNA binds first to the small subunit of the ribosome, then the large subunit comes into place, and people talk about the peptidyl transferase activity in the large subunit, which joins the amino acids together as well. So well done, you guys. Okay, let's go back and move on to the very last question. So, question 5, all about trees, elm trees. Ulmus procera, the English elm tree. Now, it got kind of wiped out sort of in the 70s by this stuff called Dutch elm disease. I remember my mum, who was around in the 70s, talked about this. She said it was a sort of catastrophe for gardeners, which she is a bit of a gardener. So fungus arrived in 1967, beetles living under the bark, spread the spores, 25 million trees were dead. That's quite a lot. So 2 reasons for the rapid spread of the fungus. Well, first of all, we've got the fact that it was spread by a vector. And there was also very low resistance to the disease, low genetic diversity. So the mark scheme has got a few other things as well. So we've got a vector that moves about, a mobile vector, an insect flying from tree to tree. I think you probably did need to get this idea that it's mobile. Low genetic diversity or lack of resistance. Fungal spores can be carried by the wind. You might have said the climate favoured the fungal growth because it was the suitable weather conditions, maybe, or suitable temperature conditions. Also, overcrowding of trees. They were planted close to each other. In fact, elm trees quite often used to be planted in these kind of rows, and they were actually produced, I think, from cuttings. So they were all genetically exactly the same clones of trees in rows that facilitated the spread. You can see the next mark here. The next one is that both of these diseases are transmitted by vectors, okay? So the Dutch elm has the beetle, which spreads the spores, and malaria has the mosquito. Here, the pathogen that causes malaria is called... Oh, it's gone. What is it called? The pathogen that causes malaria has got a plasmodium there. It is plasmodium. There's a couple different types of plasmodium. Plasmodium falciparum, Plasmodium vivax, and there's another one as well that all cause malaria. This organism belongs to the kingdom. Remember, the 3 kingdoms are Archaebacteria, Eubacteria, and Eukaryota. So it's Eukarya. And the pathogen that causes malaria and Dutch elm disease are both in the domain that is... Oh, I've got those the wrong way around. Sorry, kingdoms, that's the 5 kingdoms. So this is Protoctista. And this one is Eukarya. Check the mark scheme for that one. Yeah, Protoctista or Protista. Basically, but not protozoa, okay? That's like, we don't use that term anymore. So it's not protozoa. eukaryota or eukarya for the last one. Final bit. Explain how a malarial parasite is able to bypass the body's primary defences. The key thing here is, what is the body's primary defences? So primary defences are things that prevent pathogens from getting into your body. What primary defence does malaria bypass? Well, it bypasses basically the skin, because mosquitoes directly inject the pathogen into the bloodstream, and that's what you have to say. Here's the mark scheme. Pathogen injected directly into the blood. Notice you have to say blood or bloodstream to get the mark. It's underlined. And you have to get some idea of injecting or piercing, okay, from the mosquito. Okay, so that is the end of that question paper. Once again, I think the most important thing that some people sort of didn't quite get right is on those 2 6-mark questions. Each of those 6-mark questions had a very clear, if you looked at the question, set of things it wanted you to do. It wanted you to talk about, for example, pressure and valves and chambers at all of those 4 letters. Or it wanted you to talk about the conclusion that was made by looking at the data and the methods and the assumptions. So that's the key message from this one. Plan the structure of your 6-mark questions. Okay, so that's the last exam run-through video I'm going to do before the summer holidays. So have a great summer holidays, and I'll see you next time.